Okay, now let's talk about Intellobacteriaceae. So what really is the Intellobacteriaceae? Intellobacteriaceae, these are gram-negative loads. These are gram-negative loads bacteria. Okay, in the previous video, we considered the structure of the gram positive and gram negative. Okay, and gram negative. So now these gram negative bacteria, which is the gram negative load, it has got the following characteristics. So let's consider the characteristics of the gram negative load. So it is catalase positive. Okay, catalase positive. What does it mean? It means that it produces an enzyme that detoxifies hydrogen peroxide into water and oxygen. Okay, so all the gram negative loads, they are catalase positive. So remember, whenever you are talking about any type of the gram negative load, you should always remember to mention these general features, because these features each gram positive negative bacterial load has. Second, gram negative bacteria, it is oxidase negative. Okay, oxidase negative. What does that mean? It means it doesn't produce the enzyme that catalyze the production of cytochrome C. Cytochrome C. It doesn't produce the enzyme that produces cytochrome C. Okay, so that's what it means, oxidase negative. So it is also able to ferment glucose. So all the gram negative load are able to ferment glucose, but some they are able to ferment both glucose and lactose here, both glucose and lactose. So this type of Intellobacteriaceae that are able to ferment lactose, we have the Intellobacter, we have the E. coli, and Klebsiella. These are able to ferment lactose and also glucose. Glucose, it is just the general sugar that is fermented by all Intellobacteriaceae. But when it comes to lactose, there is exception. So exception, which one doesn't ferment lactose? We have Salmonella, Shigella, Proteus, and also Yersinia. Another thing, it is all gram negative logs. There are loads. There are facultative anilobe. Facultative anilobe. What does it mean? Meaning it can require oxygen to glow or no oxygen for it to glow. Meaning it can glow on both, whether there is presence of oxygen or absence of oxygen. That's what it means, facultative anilobe. All the gram negative can be cultured on blood agar, okay, also McConkey agar and chocolate agar. These bacteria can be cultured on these agar. Some have flagella and others they do not have flagella. There are also non-spore formations. Non-spore formation. They produce the type of toxin known as endotoxin. Just we said for the gram-negative and for the gram-positive in the previous video that the gram-positive bacteria produces the exotoxin, but for the gram-negative it produces the endotoxin. Which molecule specifically? Which part? It is the lipoprotein. Now the specific components of the lipoprotein which produces endotoxin, it is the lipid A. The lipid A. Remember the lipopolysaccharides consists of the O antigen, lipid A, and the lipopolysaccharide. But now specifically the lipid A, it is the one that produces the endotoxin. The endotoxin. So another thing, just like you have said here, the lipopolysaccharide, it has 3 molecules, which is O antigen, copolysaccharide, and also the lipid A. Also, all the gram-negative loads, it is made up, it has the type 3 circulation protein. Type 3 circulation protein. What is this protein? This protein is like a syringe that injects the endotoxin into the host. So the gram-negative loads use this type 3 secretion protein to inject the toxins into the hosts. Then these toxins produces now the pathological effect to the host cells. The infections, the way the Intellobacteriaceae can cause infection is either by injecting toxins or by itself entering the cells, or just by injecting the toxins. Let us now consider the general virulence factors of the Intellobacteriaceae. So this is the general virulence factor. This means that every Intellobacteriaceae have these factors that contribute to causing an infection. We have the capsule. The capsule also, it is called the slime layer. So the capsule, it contains the K antigen, which is the virulence factor. This is K antigen, which is also refers to VI antigen. When you consider the lipopolysaccharide, it has the virulence factor, which is the lipid A. This lipid A, it produces the O antigen. Then also, to those Intellobacteriaceae, it is the exception, meaning those which doesn't have the flagella. Those with no flagella can produce the H antigen. In the flagella, there is the H antigen. Okay, this H antigen, it is also very dangerous, which causes the pathological effect or contribute to infection. And also the type 3 secretion protein or system, it is also the virulence factor, because this is the syringe, the injector that the gram-negative rod uses in order to inject its toxins into the host cell. So these are the examples of the Intellobacteriaceae, meaning the common pathogenic Intellobacteriaceae gram-negative rods. We have the E. coli, the Klebsiella, we have the Citrobacter, we have the Celecia, we have Salmonella, we have Shigella, Proteus, Yersinia. But now there are these, the one that I've put in red and the one that I've put in black. The one that I've put in red, these are the gram-negative rod that ferment lactose. That what it ferments lactose. These are lactose fermenters. But the one in black, they are non-lactose fermenters. Okay. But what you need to know, when it comes to fermenting glucose, all the Intellobacteriaceae, they ferment glucose. So red color, lactose fermenter, then black color, non-lactose fermenters. Now let us consider the common infections that can be caused by negative rod. The first one, the Enterobacteriaceae can infect the central nervous system, and one of the pathogens that can infect the central nervous system, it is the E. coli. It also responsible for infecting the respiratory tract. So like organisms that are responsible for that are Intellobacter, E. coli, and Klebsiella. So what you need to know about E. coli, E. coli is very pathogenic. It's very dangerous. It is the pathogen that causes a lot of infections in different body organs. It also, Enterobacteriaceae can also enter the bloodstream and cause bacteremia and sepsis. Bacteremia simply means the presence of the bacteria itself in blood. Then sepsis simply means, septicemia simply means the bacteria producing toxins in the blood. With this can lead to fever. Then when we talk about another, it causes infection in the GIT tract, gastrointestinal tract. So an example, we have Salmonella, Shigella, E. coli, Yersinia, and also UTIs, Klebsiella, E. coli, Citrobacter, Proteus. One can get the gram-negative rod through fecal contaminated, oral fecal contaminated. It can be food contaminated with fecal, or it can be water contaminated with fecal, or even fingers can. Also person to person, one can get the infection. Also, Intellobacteriaceae, they are the normal flora. They are also the normal flora of the body. So these normal flora can cause infection when it leaves its place of inhabitants and goes to the foreign location. For example, E. coli, which is the normal flora of the intestine, when it goes to the blood, it can cause infection there, which is bacteremia, septicemia, and also when it goes to the central nervous system, it can cause meningitis and causing the UTI. The next one, it can be through nosocomially. This simply means hospital-acquired infection. So nosocomial simply means hospital-acquired infection. So, and the organisms that are responsible for that, Klebsiella, Intellobacteriaceae. Now let's just talk about the 4 Intellobacteriaceae. These, they are less common to cause the infection, but when they cause infection, it mostly causes the UTIs infection. So Proteus, Enterobacterius, and also Citrobacter mostly cause UTIs. Let's talk about now Klebsiella. There is 2 types of Klebsiella, which is the Klebsiella pneumoniae, okay, and also Klebsiella oxytoca. The most common type of Klebsiella that causes most of the infection, it is Klebsiella pneumoniae. This Klebsiella pneumoniae, it is commonly known as the main causative for pneumonia. So this can infect the respiratory tract, and it can also cause the UTIs. What are the virulence factors? The virulence factors of Klebsiella, it is just the common virulence factor for the gram-negative load. Just like we mentioned, capsule. The capsule contains the K antigen, okay, antigen, and also the lipopolysaccharide, which has the lipid A and O antigen, okay. Then also the type 3 secretion system, which is just the syringe the gram-negative load uses to inject its toxin. Now let us talk about the Escherichia coli. Escherichia coli, it is the gram-negative load, okay. It is the gram-negative load. What are the characteristics of Escherichia coli? All what you need to know is all the gram-negative loads, they have the general characteristics of which we also discussed. Catarrhs positive, oxidase negative. There are glucose fermenters, and some are lactose fermenter. So example of lactose fermenter Intellobacteriaceae, it is the same E. coli. So E. coli, it is a lactose fermenter. non-spore formation. There are facultative Antelope, meaning that it can globe both in oxygen presence and oxygen absence. So these are the general characteristics. We can also consider the general virulence factors of the Intellobacteriaceae, which is the lipopolysaccharides, the capsule, the type 3 secretion system. Okay, so this is the general characteristics of the Intellobacteriaceae. So now, since it is the general characteristic, it also means that it is the characteristic of Escherichia coli. So these are the virulence factors for the Escherichia coli. There is the lipopolysaccharide. This lipopolysaccharide, it contains 3 molecules, okay? The first one, it is the O antigen. Second one, it is the co-lipopolysaccharide. Then the last one, it is the lipid A. So this lipid A, it is responsible for producing endotoxins. These endotoxins, they cause damage to the cells. And also, Escherichia coli, it has got another virulence factor, which is the H antigen. H antigen is produced from the flagella. So flagella, does not only help the bacteria to move, but it releases the virulence factor. Another thing, it is the capsule. Capsule, it also produces another antigen, which is the K or VI antigen. Escherichia coli has also another virulence factor, which is the type 3 secretion system. This type 3 secretion system, it is the syringe the Escherichia coli uses to inject its toxins. The features of the Escherichia coli, which is also the general feature for the gram-negative rod. And these, they are the virulence factors. The general virulence factor for the Escherichia coli species. There are 5 types, of which we are going to consider them separately. To start with, let's just give the overview. There is the Entelopathogenic E Coli, okay, and also there is the invasive E. coli. Okay, there is the Endotoxigenic E. coli, there is Entelloaggressive E. coli, and the Interorigic E. coli. These are the 5 types of E. coli. And these 5 types of E. coli, they all share the general virulence factors. But each one of these has the specific endotoxin. The specific endotoxin that is produced by the lipid A, that is not shared by this E. coli or this E. coli or this E. coli or this E. coli. But both, remember, they have got this general virulence factor. So the characteristics we have talked about, which is they ferment lactose and glucose, catalase positive but oxidase negative. They can be cultured using blood agar, chocolate agar, and also McConkey agar. There are also the facultative Antelope, and they have the virulence factors such as the capsule lipopolysaccharide, and the flagella. This is the general virulence factor, which when you go into capsule produces the K antigen, the lipopolysaccharide, it has got the lipid A, which produces the endotoxin, which this endotoxin, it is specific to each E. coli. Endotoxin, it is just the general, which have different groups of toxins, specific toxins. So, and also we have the flagella, which can have the H antigen. Let us now consider the first type of the E. coli, which is the Entellotoxigenic E. coli. So this Entellotoxigenic E. coli, it has these virulence factors. So remember, it has these general virulence factors, which is the H antigen, O antigen, and also it has the endotoxin produced by the lipid A. It has also the K antigen and also the type 3 circulating system. Okay, so now these are the endotoxins, which are specific to it. We have the EAT label and also the heat stable. The EAT label and the EAT stable, which is EAT stable LT. So this is the virulence, this is the endotoxins that is the lipid A produces. It has also adhesing molecule, which is known as colonization factor adhesing. This is the adhesive molecule. It helps the ETEC to bind to the epithelial cell and cause the pathological infections, effects. What are some of the clinical features? What type of infection? Where mostly does it manifest? So it causes watery diarrhea. Okay, causes watery diarrhea, mostly common in children and traveler. So it causes traveler's diarrhea and also diarrhea in children. This type of diarrhea, it is similar to that of cholera, but this one is mild. It's not severe as cholera can be. How is it transmitted? Just know that all the Intellobacteriaceae can be transmitted through oral fecal contacts. It can be the water being contaminated. It can be the food or finger. Okay, let us talk about the pathogenesis. Well, the pathogenesis of the E. coli, specifically the ETEC, it starts when the adhesive molecule binds the epithelial cells of the small intestine. This adhesive molecule, it is known as colonization factor adhesin. It binds to the epithelial cell of the small intestine. After binding, it uses the type 3 system to inject its toxin. What is the name of its toxin? There are two types of the toxins. It is the heat labile and also the stable, which is the heat stable. So when it injects this toxin using this syringe, it causes the activation of guanylate cyclase. This guanylate cyclase, what does it do? It increases the production of cyclic GMP. What does cyclic GMP does? Cyclic GMP, it also activates the adenylate cyclase. Then adenylate cyclase, it increases the release of cyclic AMP from the ATP. So cyclic AMP is being produced from the breakdown of ATP. So the increase of cyclic AMP, it leads to the production of or the release of ions such as the sodium ions, chloride ions, and also water secretion. So this leads to watery diarrhea. Watery diarrhea. So heat labile toxins stimulate the production of cyclic AMP. Then heat stable, it stimulates the production of cyclic GMP. The second type of the E. coli, which is the EPEC. EPEC stands for Intellopathogenic E. coli. What is E. coli? Intellopathogenic E. coli. So what is the characteristics of EPEC? Same characteristics of the E. coli, which is catarase positive, oxidase negative, ferment lactose and glucose. They have the lipopolysaccharide, which is the virulence factor, the flagella, which contains the H antigen, the capsule, which produces the K antigen. It is also facultative Antelope. So these are the general characteristics. What are the virulence factors? Remember also to put the general virulence factors, which is the O antigen, H antigen, K antigen, the lipid A, which produces the endotoxin. Lipid A, which produces the endotoxin, and it has the type 3 secretion system, which is the syringe of the E. coli species. This type 3 system, it is the protein that every E. coli has. So now, what is the adhesive molecule? So adhesive molecule of the Entelopathogenic, it is a bundle-forming pili. This is the bundle-forming pili. Bundle-forming pili. Bundle-forming pili, which is abbreviated as BFP. When it binds, then it uses its syringe to inject the two types of protein into the cell. The first is the translocated intimin receptor. Translocated intimin. It is the translocated intimin. It is being injected. Then another one, it is the E. coli secreted protein, which are proteins which are very pathogenic to the host cell. What is the clinical manifestation? It is the watery diarrhea, but commonly in infants. How can someone get the infection? It is through oral, fecal, and also person-to-person contact. Remember that Intellobacteriaceae infections are very much common in developing countries. Very much common in developing countries. Let us now talk about the pathogenesis. It also starts with the bacteria sticking to the epithelial wall of the small intestine. What name of that adhesive molecule? The name of the adhesive molecule, it is the bundle-forming pili. This bundle-forming pili, when it sticks, then it uses type 3 secretion protein to inject what? The two types of protein into the endothelial cells. So these two types of proteins, it is known as the translocated intimin and also the E. coli circulation protein. E. coli and also E. coli circulated protein, which attacks, what does it do? It attacks the cytoskeleton of the villi. After attacking the cytoskeleton of the villi, this leads to malabsorption and water diarrhea. Okay, so this is how the pathogenesis of the EPEC is. And remember that all the Intellobacteriaceae can be identified through culturing. Culturing is the main laboratory procedure that is done in order to identify all Intellobacteriaceae species. Let us now consider the EHEC, which is Interorigic E Coli. So remember the first two that you have talked about, which is the ETEC and also the EPEC. it causes the watery diarrhea. Now here, just by the word itself, hemorrhagic E. coli, what do you think can it cause? It is the bloody diarrhea. It is the blood diarrhea. And it produces the variance factor that is similar to what Shigella produces, which is the Shiga toxin. This is the endotoxin that the lipid A of the hemorrhagic E. coli produce. Okay, then this is the serological identification, which is O157H7. So this is the serological identification of the E. coli, which is hemorrhagic E. coli. So when you talk about the virulence factor, remember always include the general one, O antigen, lipid A, which produces this same toxin, which is the Shiga toxin, H antigen, K antigen, and also the syringe system, which is the type 3 secreting system. So now let's talk about now the clinical features. 1, bloody diarrhea. It can also called hemorrhagic colitis and hemolytic uremic syndrome, not hemorrhagic but hemolytic uremic syndrome. It does produce the hemolytic uremic syndrome when its Shiga toxin 2 enters the epithelial cells of the glomerula of the kidney. Then it brings about the hemolytic uremic syndrome. How is it transmitted? It is also holofecal transmission and also person to person. Let us now talk about the pathogenesis. This pathogenesis starts with the cell, the bacteria, to adhere to the epithelial cells of the colon, okay, of the colon. So remember the type of E. coli that causes blood diarrhea, it affects the colon, and those that causes non-blood diarrhea, it is the small intestine. That's where it causes damage. Then when it adheres to the epithelial cells of the colon, it uses type 3 secretion to inject the Shiga toxin. What does this Shiga toxin does? The Shiga toxin, it disrupts the protein synthesis of the cells by binding or sticking to the RNA, which is ribosomal RNA, thereby inhibiting protein synthesis. When the protein synthesis are inhibited, this leads to the damage or the cell to die. Okay. And when it goes to the glomerular endothelial cells, the Shiga toxin 2 specifically, it causes damage and hence causes hemolytic uremic syndrome. Let us consider the next one, which is the fourth, which is the invasive E. coli. Just by the word itself, invasive, this is the only Intellobacteriaceae that enters the epithelial cells. It does not only use its toxin to inject, but itself enters the cell and cause the infection. So because it enters the cell, it can also cause blood diarrhea. It can also cause blood diarrhea. What is the general characteristics? Same, just the general characteristics for the Intellobacteriaceae, which can be the one, catalase positive, oxidase negative, ferment lactose, ferment glucose, facultative anaerobe, and also it is gram-negative load. So when we talk about the virulence factors, the general virulence factors, which is the lipid A, which O antigen, which is also the H antigen, which is also the K antigen. So these are the general for the all E. coli species. Then, not only E. coli species, but the Intellobacteriaceae. But for those which doesn't have capsule and those which doesn't have the flagella, do not produce the H antigen and the K antigen. But since all the gram-negative bacteria as the lipopolysaccharide, they all have the O antigen and the lipid A as the virulence factors. So now, what is the virulence factor? I mean the endotoxic. What is the endotoxin that is produced by the lipid A? It is the hemolysin A. Hemolysin. Just from the word itself, hemolysin, this simply means it lyses the red blood cells, hence leading to blood diarrhea. Then it has the the adhesive protein, which is invasive plasmid antigen. It enters the small intestine by itself, which is similar to the sugar pathogenesis. So the pathogenesis of the invasive E. coli, it is similar to that of the Shigella. What is the clinical features? It can cause dysentery. Dysentery means diarrhea with blood, which is just bloody diarrhea. It can cause white blood cells to manifest in stool and also bloody, that's like I said. So now, what is the pathogenesis? One, it first attaches the colony using the plasmid antigen. Then after binding to the epithelial lining, it causes the destruction of the epithelial lining, okay? Then it is being phagocytosed by the macrophage that is found in the M cells of the intestine, the intestinal lining. Then inside the macrophage, it escapes the destruction of the macrophage and it moves out of the macrophage. Then what it does it do? Then it invades, it attacks the adjacent cells. When attacking, it damages the cells, injecting its toxins, which is the hemolysin A. Injecting the hemolysin A within the adjacent cells causing damage which can lead to abdominal pain, loss of water loss, hence watery blood diarrhea. So this is the Intellobacteriaceae. If you like the video, please subscribe and share it to your friends so that they too can benefit, and comment down and also like the video. Thank you.